day students. Today we shall see a new topic, sequence analysis in bioinformatics. Our first topic for the day would be pairwise sequence alignments. Basic concepts of sequence alignment. Sequence comparison is an important method for the analysis of structure and function of biological macromolecules such as proteins, DNA or RNA. Mutation and selection during evolution over millions of years can result in considerable divergence between present-day sequences derived from the same ancestral gene. Substitutions, insertions and deletions of the bases at the corresponding positions of the ancestral sequence occur during evolution as a result of point mutations. These changes cause sequence to diverge from the common ancestor and can also lead to changes in the sequence length. As a result of mutation, even the sequences of the same protein or gene from two closely related species are rarely identical. Events like fusion of sequences from two different genes and gene duplication also occur in genomes. It is presumed that present-day homologous sequences have diverged from a common ancestral sequence through iterative molecular changes. Sequence alignment enables the researcher to determine whether two sequences exhibit sufficient similarity and thus draw a conclusion that two aligned genes share a common evolutionary history and are homologous. Alignment indicates the position of insertions and deletions and show us which bits of a sequence are variable and which bits are conserved. An alignment helps us to study the following. Compare nucleic acid and protein sequence to determine the functional, structural and evolutionary relationships between them. The degree of sequence conservation in an alignment which reveals evolutionary relatedness of different sequences. Identification and quantification of conserved regions or functional motifs. Identification of pseudogenes, profiling of genetic diseases. Phylogenetic analysis, ancestral sequence profiling and prediction, and compare a query sequence to that of a sequence present in a database or commonly called as database search. Information generated from sequence alignment can also help in assigning functions to unknown proteins, determine the evolutionary relationship of organisms, helps to predict the 3D structure of a protein, homopolymeric stretch profiling, identification of genetic disease and ancestral sequence profiling, etc. Pairwise alignment. Among the different comparison approaches, sequence alignment is the most fundamental process. It is a procedure of comparing two pairwise alignment in which one attempts to infer which positions within the sequence are homologous, that is, which sites share common evolutionary history. Here sequences are compared one another, searching for common character patterns and a residue-residue correspondences are established among related sequences, preserving the order of the residues within the compared sequences. Comparing one sequence onto another is termed as pairwise alignment and the degree of similarity between the two is quantified using a scoring function. The simplest approach is to line up the sequences against each other and insert additional characters or gaps to bring the two strings into vertical alignment as in figure 1 and score the alignment by counting how many positions match identically at each position using a predefined scoring function. The one with the best score would be considered the best alignment. For example, using a scoring function, let us set the benefit of a match to +1, the cost of a mismatch to -3 and cost of aligning a character to a gap to -4. Figure 1 shows two possible alignments of two sequences. In the first potential alignment, there are 8 matches, 8 into +1 which is equal to +8. One mismatch, 1 into -3 equals -3 and one site aligned with a gap 1 into -4 equals -4 for a total score of +1. In the second potential alignment, there are 5 matches +5, 4 mismatches -12 and one site aligned with a gap -4 for a total score of -11. In this elementary example, the score increases when more identical residues have been aligned. Conversely, the process of alignment can be measured in terms of the number of gaps introduced and the number of mismatches remaining in the alignment. A metric relating such parameter represents the distance between two sequences. That is, the measured terms like number of gaps and mismatches will be smaller if the two sequences in comparison are evolutionarily closely related. In the ideal case, in which a sequence alignment genuinely reflects the evolutionary history of two genes or proteins, residues that have been aligned but are not identical would represent substitutions. Regions where the residues of one sequence correspond to nothing in the other would be interpreted as either an insertion into one sequence or a deletion from the other. These gaps are usually represented in the alignment as consecutive dashes or other punctuation character aligned with letters. Figure 2 shows an alignment between the homologous trypsin protein from mus musculus or house mouse and Astrakus astrakus, the broad-fingered crayfish, from which it can be calculated that these two sequences have 41% identity. Conserved positions are often of functional importance. Alignment of trypsin proteins of mouse Swiss prot P07146 and crayfish Swiss prot P00765. Identical residues are underlined. Indicated above the alignments are 3 disulfide bonds with participating cysteine residues conserved, amino acid side chains involved in the charge relay system with asterisk mark, and active side residue governing substrate specificity with diamond mark. In a residue by residue alignment, it is often apparent that certain regions of a protein or perhaps specific amino acids are more highly conserved than others. This information may be suggestive as to which residues are most crucial for maintaining a protein's structure or function. In the trypsin alignment of figure 2, the active site residues that determine substrate specificity and provide the key residues of serine proteases correspond to conserved positions, as do the cysteine residues that form several disulfide bonds important for maintaining the enzyme structure. On the other hand, there may be other positions that do not play a significant functional role yet happen to be identical for historical reasons. Sequence alignments provide a useful way to gain new insights by introducing structural and functional properties of a novel protein from comparisons to those that have been well studied. The overall goal of pairwise alignment is to find the best pairing of the sequences such that there is maximum correspondence among residues. To achieve this goal, one sequence needs to be shifted relative to the other to find the position where maximum matches are found. It is impossible to evaluate all possible alignments. Take the simple case where a sequence of 100 characters is being aligned with a sequence of 95 characters. If we do add 5 gaps to the second sequence to bring into 100 total sites, there are approximately 55 million possible alignments because we may need to add gaps to both sequences and the actual number of possible alignments is significantly greater. Scoring and alignment. Since it is possible for two sequences to be aligned in a variety of different ways, the best possible alignment for any given pair of sequences is done by calculating a numerical value or score for the overall similarity of each possible alignment so that the alignment can be ranked in some order. The alignment giving the best score is referred to as the optimal alignment. The best scoring alignment will not necessarily be the correct one, and conversely that the correct alignment will not necessarily have the best score, since no scoring scheme is perfect to model the complex evolutionary process. A scoring scheme can either measure similarity or difference, the best score being a maximum in the similarity and a minimum in the difference scheme. The choice of a scoring function that reflects biological or statistical observations about known sequences is important to produce good alignments. Protein sequences are frequently aligned using substitution matrices that reflect the probabilities of given character-to-character substitutions. A series of matrices called PAM matrices, point accepted mutation matrices, explicitly encode evolutionary approximations regarding the rates and probabilities of particular amino acid mutations. Another common series of scoring matrices known as BLOSUM, block substitution matrix, encodes empirically derived substitution probabilities. Variants of both types of matrices are used to detect sequences with differing levels of divergence. The simplest way of quantifying similarity between two sequences is percentage identity, which describes the degree to which two or more sequences are actually at each position and is simply measured by counting the number of identical bases or amino acids matched between the aligned sequences. Percentage identity is obtained by dividing the number of identical matches by the total length of the aligned region and multiplying by 100. Since there are only 4 different nucleotides in nucleic acid sequences and only 20 different amino acids in protein sequence, there is always a small but finite probability for any aligned sequence that identical residue will be matched at some position. Unrelated sequences are expected to align matches at several positions, as there are often hundreds of residues in a protein sequence and thousands in a nucleotide sequence. Therefore, the length of the sequence is also to be considered. A 30% identity over a long alignment is less likely to have arisen by chance than a 30% identity over a very short alignment. Significance of an alignment is therefore assessed by statistical methods. Methods of sequence alignment. Alignment of two sequences is performed using the following methods: dot matrix analysis, the dynamic programming algorithm, word or k-tuple methods such as used by the programs FASTA and BLAST. Dot matrix analysis. Dot matrix analysis, first described by Gibbs and McIntyre in 1970, is primarily a method for comparing two sequences to look for possible alignment of characters between the sequences. It can readily reveal the presence of insertions or deletions and direct and inverted repeats in protein and DNA sequences that are more difficult to find by the other more automated methods. This method can also be used for predicting regions in RNA that are self-complementary, which have the potential to form secondary structures. The major advantage of this method for finding sequence alignment is that all possible matches of residues between two sequences are found, leaving the investigator the choice of identifying the most significant ones. Dot matrix method displays any possible sequence alignment as diagonals on the matrix and should be used first if the sequences are not known to be very much alike. In this method, one sequence A is listed across the top of a page and other sequence B is listed down the left side as illustrated in figure 3. Starting with the first character in B, one then moves across the page, keeping in the first row and placing a dot in any column where the character in A is the same. Illustration of the manner of construction of the dot plot matrix using a simple residue identity matrix to score an X where a pair of identical residues is observed, adopted from Atwood and Parry-Smith, Pearson Education. The second character in B is then compared to the entire A sequence and a dot is placed in row 2 wherever a match occurs. This process is continued until the page is filled with dots representing all the possible matches of A character with B character. Any region of similar sequence is revealed by a diagonal row of dots. Isolated dots which are not on the diagonal represent random matches that are probably not related to any significant alignment. The plot is characterized by some apparently random dots or noise and a central diagonal line where a high density of adjacent dots, the signal, indicates the regions of greatest similarity between the two sequences. Figure 4, graphical representation of dot plots showing of two identical sequences, B, two highly similar sequence, C, two different but related sequences, adopted from Atwood and Parry-Smith, Pearson Education. Within a dot plot, two identical sequences are characterized by a single unbroken diagonal line across the plot as shown in figure 3a. Two similar sequences will be characterized by a broken diagonal. The interrupted region indicated the location of sequence mismatches. Figure 4b, a pair of distantly related sequences with fewer similarities has a much noisier plot. In this case, diagonal cluster of dots are observed parallel to the central diagonal separated by a distance. Figure 4c that represents the number of insertions required to bring the sequences into correct register. To filter out random matches between compared sequences, a sliding window of defined size can be used. Instead of comparing single sequence positions, a window of adjacent positions in the two sequences is compared at the same time and a dot is printed on the page only if a certain minimal number of matches occur. A dot matrix analysis can also reveal the presence of repeats of the same sequence character many times, as these repeats become apparent on the dot matrix of a protein sequence against itself as horizontal or vertical row of dots that sometimes merge into rectangular or square patterns. They can also be used to find direct and inverted repeats within sequences. Repeated regions in whole chromosomes can also be detected by a dot matrix analysis with the help of interactive web-based computer programs. Dynamic programming. This method compares every pair of character in the two sequences and generates an alignment. The alignment will include matched and mismatched characters and gaps in the two sequences that are positioned so that the number of matches between identical or related character is the maximum possible. Dynamic programming guarantees optimal alignment and provides important information for making functional, structural and evolutionary predictions on the basis of sequence alignment. Both global and local type of alignments are made by simple changes in the basic dynamic programming algorithm. Global alignment and local alignment. An alignment that essentially spans the full extent of the input sequences is called a global alignment. In global alignment, two sequences to be aligned are assumed to be generally similar over the entire length and tried to align all of the sites optimally within the sequences. Alignment is carried out from beginning to end of both sequences to find the best possible alignment across the entire length between the two sequences of roughly the same length. Figure 5. Optimal global sequence alignment. Alignment of the amino acid sequences of human zeta crystalline and E. coli quinine oxidoreductase. Identical residues are marked by asterisk below the alignment and dots indicate conserved residues. The trypsin and quinone oxidoreductase or zeta crystalline alignments discussed in figure 2 and figure 5 are examples of global alignments. Proteins consisting of a single globular domain and homologous sequences that have not diverged substantially can often be aligned using a global strategy. For divergent sequences which result from large-scale sequence rearrangement and genome shuffling, only subsections of the sequences may be homologous and of variable lengths, or the homologous sections may be in a different order. For example, a long sequence may be ordered A, B, C, D, E, F, where each letter represents a section of the sequence and not an individual site. A sequence inversion of section C, D, E may change the sequence in another species to A, B, E, D, C, F. Figure 6. Illustration of global alignment problem. Sequences A, B, C, D, E, F and A, B, E, D, C, F cannot be properly aligned because the homologous sections of the sequences are not in the same order. Although each section of the first sequence is homologous with a section of the second sequence, they cannot be globally aligned because of the rearrangement and fails to recognize highly similar local regions between the two sequences. Figure 6 shows possible global alignments if section C, D or E is aligned. In each case, the other two sections cannot be aligned properly. Local alignment. Global alignment strategy was developed before the exon-intron structure of genes had been discovered. Many proteins do not display global patterns of similarity, but instead appear to be mosaics of modular domains. Figure 7 shows a modular structure of two proteins involved in blood clotting: coagulation factor 12 F12 and tissue type plasminogen activator PLAT. Besides the catalytic domain, which provides a serine protease activity, these proteins have different numbers of other structural modules: two types of fibronectin repeats, a domain with similarity to epidermal growth factor, and a module that is called a Kringle domain. Figure 7. Modular structure of two proteins involved in blood clotting. Schematic representation of the modular structure of human tissue plasminogen activator and coagulation factor 12. A module labeled C is shared by several proteins involved in blood clotting. F1 and F2 are frequently repeated units that were first seen in fibronectin. E is a module resembling epidermal growth factor. A module known as a Kringle domain is denoted K. These modules can be repeated or appear in different orders. Patterns of modularity often arise by in-frame exchange of whole exons. Global alignment methods do not take this phenomenon into account. In a local alignment, subsections of the sequences are aligned without reference to global patterns of similarity over the entire length. Local alignment allows the alignment program to align regions separately regardless of overall order within the sequence length. Figure 8 shows an illustration of global and local alignment of a sequence that is not sufficiently similar but shares common regions. Figure 8. Illustration of global and local alignments of two sequences that are not sufficiently similar. Local alignment helps to align similar regions while allowing highly divergent regions to remain unaligned. It only finds local regions with the highest level of similarity between the two sequences and aligns these regions without regard for the alignment of the rest of the sequence regions. This approach can be used for aligning more divergent sequences with a goal of searching for conserved patterns in DNA or protein sequences, and can also be used for aligning divergent biological sequences containing only modules that are similar, such as domains or motifs. The rationale for local similarity search is that functional sites like catalytic sites of enzymes are localized to relatively short regions which are conserved irrespective of deletions or mutations in intervening parts of the sequence. Some of the common publicly available implementation of pairwise comparison programs like BLAST and FASTA uses local alignment strategy and are made to run even faster by incorporating heuristics. Needleman and Wunsch Smith-Waterman algorithms for pairwise alignments. Needleman and Wunsch algorithms. In this approach, a maximum match between two sequences is defined to be the largest number of amino acids from one protein that can be matched with those of another protein while allowing for all possible deletions. A penalty is introduced to provide a barrier to arbitrary gap insertions. In its simplest form, Needleman and Wunsch in 1970 proposed a maximum match pathway in which cells representing identity scored 1 and cells representing mismatch are scored 0. A 2D array similar to dot plot is populated with these values. An operation of successive summation of cells then commences. This process examines each cell in the matrix. The maximum score along any path leading to the cell is added to its present contents and the summation continues. When the process is completed, the maximum match pathway is constructed. An alignment is generated by working through the matrix, starting at the highest scoring element and the N termini and following the pattern of high scores through to the C termini. Leaps to non-adjacent diagonal cells in the matrix indicate the need for a gap insertion to bring the sequence into register, as long as the gap penalty barrier permits opening a gap at that point. If the barrier does not permit gap insertion, a lower scoring pathway may have to be taken. Needleman and Wunsch algorithm produces an alignment that takes into account all residue of the input sequence. The starting point of the maximum path tracked back is always at the N termini and is calculated from a scoring process that comes from C termini. For this reason, the method results in a global alignment. The Smith-Waterman algorithm. Consider two sequences that are only distantly related to each other. Even so, they exhibit small regions of local similarity, although no satisfactory overall alignment can be found. Smith-Waterman in 1981 described a method for finding these common regions of similarity. As in Needleman and Wunsch, this algorithm also employs a matrix-based approach, and backtracking is used to reconstruct the gapped alignments. A key feature of this algorithm is that each cell in the matrix defines the endpoint of a potential alignment whose similarity is represented by the value stored in the cell. The algorithm begins by filling the edge elements in a 2D matrix with a 0.0 values, because the cells represent the ends of alignment of length 0 and consequently their similarity score is 0. Cells in the matrix are populated with floating point values rather than integer. The next step is to populate cells in the matrix. This is achieved by evaluating 3 functions and choosing the maximum of the 3 values, or 0 if a negative value would result. These functions consider the possibilities for ending an alignment at any particular cell. First, the similarity score, example 1.0 for a match, minus 0.333 for a mismatch for the diagonal predecessor of the cell under consideration is added to that cell's score, then the maximum value is calculated for a deletion representation along A, the current row of the matrix, and B, along the current column of the matrix. Finally, if a negative score would result, 0.0 is substituted to indicate that there is no alignment similarity up to the current cell position. Once the matrix is complete, the highest score is located, which represents the endpoint of the highest scoring alignment between the two sequences, and the other elements leading to this are determined using a backtracking procedure. The essential difference between the two algorithms is that in Smith-Waterman, the matrix contains a maximum value that may not be at the end termini of the sequence. It represents the endpoint of an alignment such that no other pair of segments with greater similarity exists between the two sequences, hence a local rather than global method. Word or k-tuple methods such as used by the programs FASTA and BLAST. Word methods, also known as k-tuple methods, are heuristic methods that are not guaranteed to find an optimal alignment solution, but are significantly more efficient and faster than dynamic programming. These methods are especially useful in large-scale database searches, where it is understood that a large proportion of the candidate sequences will have essentially no significant match with a query sequence. Word methods identify a series of short, non-overlapping subsequences or words in the query sequence that are then matched to the candidate database sequences. The first widely used program which performs optimized database similarity searches for local alignments using a substitution matrix was FASTA. Since it would take a substantial amount of time to apply this strategy exhaustively to improve speed, FASTA uses the observed pattern of word hits to identify potential matches before attempting the more time-consuming optimized search. In this method, the user defines a value k to use as the word length with which to search the database. Most potential matches are then further evaluated by performing a search for a gapped local alignment. A full Smith-Waterman alignment search is performed. This method is slower but more sensitive at lower values of k, which are also preferred for searches involving a very short query sequence. FASTA provides an estimate of the statistical significance of each alignment found. The program assumes an extreme value distribution for random scores, but with the use of a rewritten form of the probability density function, in which the expected score is a linear function of the natural log of the length of the database sequence. An expectation E is calculated, which gives the expected number of random alignments with z scores greater than or equal to the value observed. BLAST. The BLAST family of search methods provide a number of algorithms optimized for particular type of queries, such as searching for distantly related sequence matches. BLAST was developed to provide a faster alternative to FASTA without sacrificing much accuracy. Like FASTA, BLAST uses a word search of length k, but evaluates only the most significant word matches rather than every word match as does FASTA. The BLAST programs introduce the idea of neighborhood words instead of requiring words to match exactly. A word hit is achieved if the word taken from the subject sequence has a score of at least t when a comparison is made using a substitution matrix to the word from the query. This strategy allows the word size w to be kept high for speed without sacrificing sensitivity. Thus, t becomes the critical parameter determining speed and sensitivity, and w is rarely varied. If the value of t is increased, the number of background word hits will go down and the program will run faster. Reducing t allows more distant relationships to be found. The occurrence of a word hit is followed by an attempt to find a locally optimal alignment whose score is at least equal to a cutoff score. This is accomplished by iteratively extending the alignment both to the left and to the right with accumulation of incremental scores for matches, mismatches, and the introduction of gaps. Interpretation of results. Sequence alignment is useful for discovering functional, structural, and evolutionary information in biological sequences. Sequences that are very much alike or similar in a sequence analysis probably have the same function. For example, similar DNA sequence will have similar regulatory role, similar protein sequences will have similar biochemical function and may have similar three-dimensional structure. If two sequences from different organisms are similar, they may have been originated from a common ancestor sequence and the sequences are then defined as being homologs. Evolution of new gene is often thought to occur by gene duplication, creating two tandem copies of the gene followed by mutations in these copies. In rare cases, new mutations in one of the copies provide an advantageous change in function. In due course, these two copies may evolve along separate pathways, although the resulting separation of function will generate two related sequence families. Sequences among both families will still be similar due to the single gene ancestor. Sequence similarity search also help in identifying several possible types of ancestor relationships like orthologs, paralogs, analogs, etc. Interpretation of alignments can also help to sort out possible evolutionary origins among similar sequences. Alignment can reveal homology between sequences. In all methods of sequence comparison, the fundamental question is whether the similarities perceived between the two sequences are due to chance and are thus of little biological significance, or whether they are due to the deviation of the sequences from a common ancestral sequence and are thus homologous. If two sequences in an alignment share a common ancestor, mismatch in the alignment can be interpreted as point mutation and gaps as indels, insertion or deletions, introduced in one or both lineages in the time since they diverge from one another. In sequence alignment of proteins, the degree of similarity between amino acids occupying a particular position in the sequence can be interpreted as a rough measure of how conserved a particular region or sequence motif is among lineages. The presence of conserved regions or only very conservative substitutions suggests that this region has structural importance. Similarly, presence of conserved base pairs indicates similar functional or structural role. Any standard alignment program will produce some statistical value indicating the probability that an alignment of a given quality could arise by chance, but does not indicate how much superior a given alignment is to alternative alignments of the same sequences. The statistics quoted in BLAST for pairwise comparisons are probability P or expected frequency E values. The P value relates the score returned for an alignment to the likelihood of its having arisen by chance is real. The closer the value approaches to 0, the greater the confidence that the match is real. Conversely, the nearer the value to unity, the greater the chance the match is spurious. The E value describes the number of hits one can expect to see by chance, in other words noise, when searching a database of a particular size. For example, an E value of 1 assigned to a hit can be interpreted as meaning that in the current search one might expect to see one match with a similar score simply by chance. Conversely, a value of 0 indicates that no matches would be expected by chance. Thus, we come to the conclusion of this session. Before we attend the next unit, please try to answer the following questions. A. How can we assess whether a given alignment between two sequences is significant or indicative of homology, common ancestry? B. Calculate the score of the DNA sequence alignment shown below using the following scoring rules: 1 for a match, -2 for a mismatch, -3 for opening a gap and -1 for each position in the gap. Here are some books for your references. Introduction to Bioinformatics by Atwood and Parry Smith, Pearson Education. Structural Bioinformatics by Philip E. Bourne and Helge Weissing, Wiley Publications. And Bioinformatics, Methods and Applications, Rastogi SC, Mendirata N. and Rastogi P., Prentice Hall of India Private Limited, New Delhi. Essential Bioinformatics, Jingxiong, Cambridge University Press. Thus, we are concluding today's session. See you next time with another unit in Bioinformatics. Goodbye.